Welcome to AP Biology. Today I want to talk about cell communication with you. So what you can see here are single-celled eukaryotes, and here's a multi-celled eukaryote. And a lot of the cell-to-cell communication that happens here actually happens in the girl too. And this is actually even true for prokaryotes with the way that they communicate. So what typically happens is one cell will release a molecule, and then another cell, maybe distant, like maybe these molecules had to go through blood to get there, or maybe they were not too far away, but this other cell might have a receptor. And if this molecule matches that receptor, then that means this one is telling that one what to do. So that's cell-to-cell communication. And it turns out that it works pretty much the same way in single-celled organisms like these, and even single-celled bacteria as well. And it works in multi-celled organisms the same way. And so that means that we have a common ancestor who had some kind of a pathway where one cell was releasing a molecule that was received by another cell with that special receptor that fit just that kind of molecule. Okay, so here's the overall view of it. Here are a couple of yeast cells, and yeast cells are fungi. They are single-celled fungi, and so here is an A-type yeast cell, mating type A, and it releases this molecule. We're going to call it A factor. It has a more generic term that we're going to give it in a little bit. But notice that the alpha mating type has receptors that fit just the factor released from the A mating type. And then the alpha type releases this pink molecule called the alpha factor, which looks more like a triangle. And of course, it doesn't really look like a triangle, but the alpha factor and the A factor really do have different shapes. And this alpha factor fits into the receptor of the A-type yeast. And so when these cells signal each other, they will recognize that the other one is around and they'll start growing toward each other. And so the method that this cell is using to communicate with this cell is very, very similar to the methods that your cells use to communicate with each other. So cells most often communicate with each other via these chemical signals. We're going to use a word called ligand to mean a small molecule that is very specific and fits in a receptor of another molecule. So biologists have discovered some universal mechanisms for cellular recognition. These are true in yeast cells, single-celled fungi. They're true in bacteria. They're true in people, right? So animals are true in plants. So here's an example. So you might be familiar with epinephrine or adrenaline. It's a hormone that makes you really stressed, right? And so the fight or flight response is triggered by a molecule called epinephrine. That is a hormone. So let's see if we can switch it to that. So I do want you to know what a hormone is. A hormone, I define it in a couple of slides, but a hormone is a molecule released by one part of your body, and if you're an animal with a circulatory system, that hormone will usually travel through the blood and get to another cell. And so when that hormone is released by the adrenal glands, it goes into the blood, and then a cell that has a receptor for it will be triggered by this epinephrine that's been released. If it's a cardiac muscle cell, it'll contract harder. If it's a muscle cell in the airways going down to the lungs, it will actually relax to open up those tubes going to the lungs. And so different cells respond differently. So cardiac muscle cells will contract harder, but smooth muscle cells will relax. So completely opposite, but to do the same thing, and that is to make you run faster to get away from whatever is chasing you. Those signaling molecules, they're very similar to what bacteria will do with each other. One bacterial cell will release molecules, they won't be hormones, but they'll be some type of ligand that will be accepted by a receptor on another bacterial cell, and that cell will do something in response. So external signals are converted to responses within the cell, and we call that transduction. Trans means across, and duction is the act of leading or bringing. So here's what it looks like. There's a ligand, which is the original small molecule. Could be epinephrine, could be serotonin, any molecule that one cell releases to affect another cell. And here's the receptor. That receptor is going to be very, very specific. And so if the ligand fits the receptor, usually the inside of the receptor here will change shape, which will cause a series of reactions to happen, which cause some kind of a response. So the transduction is how you change from the initial part to the final part. It's all these steps in the middle. So a signal transduction pathway. So a pathway, if you think about a regular pathway in English, you're just walking along some path through a park or something, right? When we're talking about this in biology, we're talking about a series of chemical reactions that happen inside of a cell. First, this molecule is changed to this molecule, then that's changed to something else, which is changed to something else, which is changed to something else. So a signal transduction pathway starts with a signal, which is um, maybe it's epinephrine, right? Maybe that's the signal that says, hey, cardiac muscle cells, you'd better contract harder. Or, hey, smooth muscle cells, you'd better relax, right? That would be the response. So the signal would be the original molecule. And then the transduction pathway are all these steps in the middle. So it's a series of steps by which a signal on the cell surface, usually the surface, is converted to a specific cellular response. So there are 3 steps. The first is signal perception. So let's say epinephrine is released by your adrenal glands, or adrenaline if you want to call it adrenaline, same molecule, released by your adrenal glands, goes through the blood, and then there's a receptor on your cardiac muscle cells, but there's no receptor on your skin. So that means the cardiac muscle cells are the targets and your skin's not. So you have to have the signal perception. If you have the ligand and there's no receptor there, or the receptor is mutated and it's the wrong shape, then nothing happens. Then there are a series of steps inside. The intracellular, intra means within the cell. Signal transduction means this original signal is transduced or changed into some response. So in this case, there are 1, 2, finally 3 steps that have to occur. Sometimes there are 10 steps, sometimes there's just a couple of steps, but there are a series of steps that happen in order to get to some type of cellular response. So the cellular response, if it's epinephrine and it's affecting cardiac muscle cells, the response is to contract harder. If it's the smooth muscle in the iris of your eye, it would be to relax. That would be the response. The response might be take in glucose and break it down. The response might be, hey, muscle cell, take your glycogen and break it down to glucose. So there are all kinds of different responses. The response might be to multiply, hey, time to do mitosis. Or the response might be, hey, we need a certain piece of DNA transcribed and translated. We need to make a certain protein, so go do that. So you need to know this series of steps here, the perception and then the transduction, the cascade of events here that gives you this signal transduction and then the response. And then I want you to be able to come up with examples of each. So I'm going to give you 3 different receptors that we'll talk about, and then sort of 4, I guess, and then different examples here, but nothing specific that you would need to necessarily memorize except maybe a few general words. And then I want you to be able to come up with examples of responses by the end of this unit.